Good morning. I am your host, Facundo Batista, and together with Richard Young and Callum Overholt, we have organized this 2021 MIT course on COVID-19, SARS-CoV-2, and the pandemic. So on behalf of the three of us, thank you for joining today. As you know, the purpose of this course is to learn what we know about this virus and the pandemic from the best scientists across the globe. From these world leaders, we have learned about the pandemic, the virus, and some immunology behind. And today we will focus on vaccines, and who better to do this than our today's speaker. I'm delighted to introduce to you the extraordinary scientist, Professor Dan Barouch. Dan is currently the William Wordsworth Castle Professor of Medicine, Professor of Immunology at Harvard Medical School, and Director of the Center of Virology and Vaccine Research at Beth Israel Deaconess Medical Center. He's also one of the founding members of the Reagan Institute. And for many years, his lab has focused on studying the immunology and virology behind HIV infection and developing innovative strategies for vaccine and disease eradication. His group has applied their vaccine expertise to preclinical and clinical studies, focusing on other infectious diseases of global significance, such as Zika virus, tuberculosis, and most recently, SARS-CoV-2. I think that it's very fair to say that Dan's efforts towards SARS-CoV-2 has been the scientific power that contributed to the developing of the single-shot J&J vaccine, which is now being rolled out in the US and throughout the world. And the reason why I show this picture is because this was the day that I took this selfie with Dan and Bruce, the day that Dan vaccine was approved by the FDA at the Reagan Institute. Dan was recently elected as National Academy of Medicine in 2020, and given his achievements, I suspect that it's going to be the first of major prestigious international recognition awards that he will get. I particularly would like to thank you, Dan, to joining us today, and given that it's a very busy time for you, and given the new variant that just appeared over the weekend. So we are delighted to have you, and thank you very much to giving this talk.
Well, thank you very much, Facundo. I'm going to share my slides. Okay, can you see my slides okay?
Yes, we can.
All right, very good. Okay, so what I'd like to do in the next 45 minutes or so is really to give you a whirlwind view of our personal journey over the last year and a half or so, and what we've been doing to help with the fight against COVID-19. In particular, I'm going to talk about some of our background on vaccine work for other pathogens, as well as for COVID-19, some of our work for all the vaccines, but our particular contributions to the J&J vaccine. So the story starts in December of 2019. A new disease emerges in Wuhan, China, a pneumonia of an unknown cause. Within weeks, the virus was identified and the SARS-CoV-2 sequence was generated and released. On January 10th of 2020, the SARS-CoV-2 sequence was released by the Chinese researchers, and on that day, there were 41 cases reported and 1 death. We actually had a lab retreat on that day, and we talked about it, and we were alarmed by the spread to this number of cases. And this was sufficient to trigger vaccine development, not just from our group, but from many, many groups throughout the world. So just think about that. Just a year and a half ago, 41 cases and 1 death were a cause of alarm. Now, instead of 41 cases, we have over 262 million cases. And instead of 1 death, we have over 5.2 million deaths. It's truly humbling to see a virus essentially take down the world and bring the world to its knees. But the scientific community has responded, and has responded in a way that is truly unprecedented in terms of the rapid understanding of the virology of this virus, the immunology, the pathogenesis of this virus, the development of public health strategies, the development of therapeutics, and of course, also the development of vaccines. The WHO has estimated that there have been more than 200 serious vaccine development programs throughout the world that really span many different platforms, including DNA, RNA, proteins, vectors, inactivated viruses, and other platforms. In the United States, we've done comparatively well with 3 vaccines approved in the US for emergency use. The first 2 that were approved, the amazing vaccines with the mRNA technology from Pfizer, now fully approved, and Moderna, hopefully soon to be fully approved, as well as the adenovirus vector-based vaccine from J&J, which also hopefully will be fully approved in the next quarter. mRNA and vector-based vaccines both belong to a class of vaccines called gene-based vaccines. And these differ from traditional vaccine modalities, which include live attenuated viruses as well as whole killed viruses. In contrast, gene-based vaccines don't actually start with a virus. They just start with the nucleic acid sequence of the target antigen. In this case, it was the COVID-19 spike protein. Earlier this year, we published a review article with our colleagues from Moderna, and we reviewed the development of mRNA and adenovirus vector technology. And essentially, there are strong parallels between the two. They both include nucleic acid for this spike protein delivered with a specialty delivery vehicle. For mRNA vaccines, it's the RNA of the spike protein, and the delivery vehicle is a lipid nanoparticle. For adenovirus vector-based vaccines, it's the DNA of the spike protein, and the delivery vehicle is an adenoviral capsid. The field of COVID vaccines took a giant leap forward on November 9th of last year, just over a year ago, when Pfizer announced the initial results of their phase 3 trial showing stunning efficacy of more than 90%. Just a week later, Moderna announced successful outcomes of their vaccine study. And in January, J&J announced successful outcomes of their vaccine study. So in the United States, we've actually completed not just 3, but actually 5 phase 3 trials of COVID-19 vaccines. Each one of these trials was huge, involving 30 to 45,000 participants. The first 2 were the mRNA vaccines of Pfizer and Moderna. The next were the adenovirus vector-based vaccines, Ad26 from J&J and ChAd-OX1 from AstraZeneca. And finally, the protein-based vaccine from Novavax. The first 3, Moderna, Pfizer, and J&J, are now approved in the US. The AstraZeneca and the Novavax vaccines are not yet approved. Vaccine development usually takes many years, if not decades, particularly for a new pathogen. So how is it possible that not one, but multiple COVID-19 vaccines could be developed in just about a year? That's truly unprecedented in the history of science and the history of vaccinology. Were these vaccines rushed? That's a question many people are asking. I would actually argue that these vaccines were not made in a year, but rather these vaccines were made over decades, because each of these vaccines reflects decades of advances in virology, immunology, and the development of gene-based vaccine platforms, particularly the mRNA platform and the vector platform. The reason why clinical development could go relatively quickly was because these vector platforms had already been tested for other pathogens. And because of the global concern and the global urgency, companies could perform development activities in parallel rather than in series, with substantial tolerance of programmatic and financial risk. For example, all companies started to manufacture millions of doses of the vaccine before the phase 3 trials were even done, a level of risk that no company would have ever tolerated in the past. But importantly, there has never been any compromise of patient safety or regulatory integrity. The initial development of the Ad26 vaccine platform actually was in our lab, and not for COVID-19 and not for coronavirus, but rather in the early 2000s for a different program, the development of an HIV vaccine. I started my own lab in 2002, and one of the first projects we decided to do is to create new vaccine platform technologies with an eye towards making an HIV vaccine. So in the early 2000s, starting in 2002, 2003, we started to develop adenovirus vector platforms. We created at least a half dozen of these. And this is the first publication on the initial cloning, vectorization, and selection of Ad26 as a candidate vaccine platform. Through our HIV vaccine program over the years, we developed Ad26 first into animal studies and then into first-in-human clinical studies. We were working with a small biotech company in Holland called Re-Challe. And then in 2012, everything changed when a large pharmaceutical company, Johnson & Johnson, acquired Re-Challe. And then for our HIV vaccine program, we suddenly had a major pharmaceutical partner rather than a small biotech company as a partner. It's an entire different lecture to talk about our HIV vaccine efforts, but I'll just say that HIV vaccines is far more difficult, and HIV presents unique and unprecedented challenges for vaccinology, and it will still be probably many years before we do have an HIV vaccine. But it's clear that without our HIV vaccine program, the Ad26 vector technology would not have been generated. We then applied Ad26 as a vector for other pathogens over the last several years. In 2016, with the emergence of Zika virus, we partnered again with J&J in developing an Ad26-based Zika vaccine. And here we show the results of a phase 1 clinical trial in about 100 individuals in which we administered either a low or a high dose of the Ad26 Zika vaccine, either as a single shot vaccine or as a 2-shot vaccine. If we look at the early responses following initial immunization, following the first immunization, then essentially everyone developed neutralizing antibodies. And in individuals that only received the single shot vaccine shown in the circles and the squares, these neutralizing antibody titers were actually relatively stable over the course of a year. A second shot of the vaccine given on day 57, which is 2 months, led to titers that were very high. And these then drifted down somewhat, but still were stable for about a year. So from the Ad26 Zika program, we learned that for a more well-behaved virus, such as Zika, compared with HIV, for example, for a more well-behaved virus, an Ad26 vector, a single shot of the vaccine could raise neutralizing antibody titers in virtually everyone. These antibodies were durable, which is not a given for other vaccine platforms, and also that a second shot of the vaccine could increase these titers to very high levels. So if we fast forward now to January of 2020, then we reasoned that the Ad26 vector platform might be able to contribute to the response to COVID-19 because this replication-incompetent common cold virus vaccine platform had already been developed for other pathogens and was therefore essentially ready to go. Ad26 HIV was the initial development of the Ad26 platform technology. Ad26 Zika demonstrated the potential of a single shot vaccine and also substantially improved responses with a 2-shot version of the vaccine. Ad26 Ebola, which was a program developed by J&J, showed that mass production and global distribution was feasible. Their formulation scientists developed liquid formulations that did not require a sub-zero frozen cold chain, which we think is very important for distribution and vaccination in the developing world. And through an Ad26 RSV program, the vaccine was administered to infants down to 4 months of age. So this is the timeline for the development of the Ad26-based COVID vaccine. On January 10th of last year, the virus sequence was released. We started working on the sequence that same evening, and we compared the sequence to the spike sequence of SARS and MERS, and we developed synthetic antigen candidates. So when we got back to the lab on Monday, January 13th, we were ready to order synthetic genes to start designing the vaccine. We signed a collaboration agreement with J&J by the end of January. And in February, we started pilot immunization studies in small and large animal models. By May, we had our first publications on basic principles of natural and vaccine immunity in a non-human primate model. Not this Ad26 vaccine per se, but rather basic research advances to try to understand immunology of COVID-19. And our hope was that these findings would be beneficial, not just for this program, but for all the vaccine programs underway throughout the world. By the end of July, we started our phase 1/2 clinical trials, and we reported the proof of concept protection of Ad26 vaccine efficacy in non-human primates. Less than 2 months later, on September 21st, we initiated the single-shot phase 3 clinical trial, and in November 16th, we initiated the 2-shot phase 3 clinical trial. And when I say we, I really mean J&J with the NIH. We're the leads of these clinical trials. By the end of January of 2021, interim safety and efficacy data was announced for the single shot vaccine, which led to FDA emergency use authorization by the end of February. By the end of this year, J&J hopes to distribute about 400 million doses of the vaccine, and by the end of 2022, this number could go up to about 2 billion. So I'll show a couple high points from a couple of the papers along the way. This was actually our first publication for SARS-CoV-2. We essentially developed the Rhesus macaque model of SARS-CoV-2 infection. And we did a very simple experiment to show that infection, natural immunity from infection protects against Re-Challe. So we inoculated Rhesus macaques with SARS-CoV-2. And on the left, you can see that there's virus replicating in the lung for about 2 weeks. And if we let these animals convalesce for a period of 35 days, and then we re-challenge them, then we have on the right much lower viral loads, a blip of virus on the first day, but really very durable, very, very robust protection. This was one of the early demonstrations of natural immunity. And at that point in time, it really was not at all clear whether humans would have natural immunity against coronaviruses or not. So when we saw these results, then we immediately became optimistic about the prospects of success for vaccines, because we know from our HIV work that the development of a vaccine for which there is no natural immunity in humans is much more challenging. By July, we reported the proof of concept data of Ad26 vaccine protection in the Rhesus macaque model. And these are the results from our challenge studies in macaques. We actually tested 7 different versions of the vaccine. The version that ultimately made it into human clinical trials included a diproline mutation that's initially reported by Jason McClellan and colleagues. And here in sham control monkeys, following challenge, there was high levels of virus replication in the bronchoalveolar lavage and lung, followed by resolution in about 10 days, and very robust protection in the vaccinated animals. We also saw very robust protection in nasal swabs, although there's always more blips in nasal swabs compared with bronchoalveolar lavage. Because we did test 7 different versions of the vaccine, some actually didn't work out so well. And so we had a spectrum of results, and we could look at immune correlates. And this is one of the early immune correlates analysis that demonstrated that the neutralizing antibody titers induced by the vaccine correlated inversely with protection, here as measured by peak viral loads in bronchoalveolar lavage. So through the vaccine studies, we could demonstrate quite reproducibly that neutralizing antibody titers were a statistical correlate of protection. And similarly, we reasoned that even in large phase 3 human clinical trials, the best we could get for correlates would be statistical associations. But we wanted to use the animal model to try to do mechanistic studies to get a deeper understanding of the mechanistic correlates of protection. So the first experiment that we did was a classic IgG adoptive transfer study. We wanted to determine if purified IgG from convalescent macaques could protect naive animals, and if so, could we define neutralizing antibody thresholds for protection. So essentially, we previously demonstrated that antibody responses correlated with protection. Here we wanted to show that we wanted to prove that antibody responses were sufficient for protection. So we took a group of convalescent re-challenged macaques that had high antibody titers. We purified IgG from plasma, and then we adoptively transferred the purified IgG into 12 naive recipient macaques 3 days prior to challenge. The first group received a high dose of 250 mg per kg. The second group received a middle dose of 25 mg per kg. A third group received a low dose of 2.5 mg per kg, and a fourth group was a sham control group. 3 days after adoptive transfer, on day 0 of the study, we challenged these animals with SARS-CoV-2. Here are the neutralizing antibody titers following adoptive transfer. Remember, the adoptive transfer occurred on day -3 and the challenge on day 0. So the neutralizing antibody titers on day 0 fully reflect the infused antibodies. In the high dose group on day 0, neutralizing titers were about 500, and over a 14-day post-challenge follow-up period, these drifted down over time. In the middle dose group, that received a 10-fold lower dose of antibodies, there was actually exactly a 10-fold lower neutralizing titer of about 50. These also drifted down over time, although one animal essentially looks like it's ticking upwards by day 14. In the low dose group of 2.5 mg per kg, these animals essentially had very low or undetectable neutralizing titers on the day of challenge, but all three of these animals had a sharp uptick of neutralizing titers on day 14 following challenge, very similar to sham controls, essentially indicative of natural infection. So here are the viral loads in bronchoalveolar lavage. In the top dose group, 3 out of 3 animals were protected. In the mid-dose group, there was 1 breakthrough. In the low-dose group, all 3 animals were infected, but there's a suggestion that virus is more rapidly controlled than in sham controls. In nasal swabs, we saw a very similar outcome. In the top dose group, there was complete protection. In the mid-dose group, there was 1 breakthrough. In the low-dose group, all the animals were infected, but these animals had lower peak virus loads and a more rapid resolution of virus compared with sham controls. So these data essentially prove that antibodies alone are sufficient for protection as long as they're above a certain threshold. The next experiment we did was a CD8 depletion study. We wanted to determine the role of CD8 T cell responses in protection against SARS-CoV-2. Now, you might say, well, why do we do this experiment? I just showed you that antibodies alone can protect. But we reasoned that antibodies might wane over time, and although we didn't really think about it because this was last fall, now we know that variants may emerge that might be partially resistant to antibodies. So in this experiment, we used a slightly different model. We wanted to determine the role of CD8 T cell responses in protection against SARS-CoV-2 re-challenge in convalescent macaques, because for those, we had more control over the antibody titers. So we infected 10 animals and we allowed them to convalesce. We watched them for a period of about 2 months and we watched their antibody titers drift downwards. Then we depleted CD8 T cells with an infusion of an anti-CD8 alpha monoclonal antibody in 5 animals, and 5 other animals received infusion of a sham antibody. So the sham antibody infused animals, we would expect to have natural immunity protection similar to what I showed you before. And the 5 animals that received a CD8 depleting antibody would be the experiment to see the role of CD8 T cells. Now, for the aficionados in the audience, you might know CD8 alpha also depletes NK cells. And so in the paper, we actually did a second experiment with an anti-CD8 beta monoclonal antibody with similar results. So animals received an antibody infusion 3 days prior to challenge, and on day 0 of the study was the challenge day where we performed a SARS-CoV-2 re-challenge in those animals. Here are viral loads following the re-challenge. In both the sham and the CD8 depletion group, we saw very good protection, although there was 1 animal that had a blip with CD8 depletion. But protection in nasal swabs is more difficult to achieve than protection in the lung. And in nasal swabs, we saw something different. There was 1 animal that showed a blip in the sham control group, but 5 out of 5 animals that were depleted of CD8 T cells showed substantial virus in nasal swabs following re-challenge, demonstrating quite directly that CD8 T cells play a role in protection. So here are peak viral loads in nasal swabs in the animals that received a sham depleting antibody, showing a very good natural immunity. If we CD8 deplete the animals, then we see about a 2 to 2.5-fold increase in median viral loads in nasal swabs. But these are still low. These are still 2 logs lower than viral loads in naive animals that are infected. So overall, this paper demonstrated several points. First, that purified IgG can protect macaques against SARS-CoV-2 challenge in a dose-dependent fashion. Second, that threshold neutralizing antibody titers for protection are relatively low, about 50 in this model. And this paper was actually, the first draft of this paper was written prior to the November 9th announcement by Pfizer. And we wrote in this paper that we thought these titers would be readily achievable by vaccination. So another reason for optimism. We also show that CD8 depletion reduced protection against re-challenge in convalescent macaques with waning antibody titers. So our overall conclusion was that our data suggests that antibodies alone can protect, but that cellular immune responses contribute to protection when antibody titers are borderline or subprotective. And I think that this model has largely held up over the past year in terms of our thoughts of immunology of SARS-CoV-2 infection. And I think this might be reinterpreted in the setting of waning antibody titers, as well as the emergence of variants that can variably evade neutralizing antibodies as well. So in terms of clinical development, then starting in July of 2020, then the phase 1/2 studies were underway. The phase 1/2 studies were a combined study involving about 800 individuals, and these were randomized into 5 different groups, receiving the placebo, a single immunization of the 5 × 10⁻⁻ viral particle dose, 2 immunizations of the 5 × 10⁻⁻ dose, a single immunization of the 10⁻11 dose, and 2 immunizations of the 10⁻11 dose. The 2 doses were very similar. Following a single shot vaccination, the majority of individuals had neutralizing antibody titers on day 29, but actually these continued to go up a bit to day 57 and day 71. So already we're seeing relatively good durability of the vaccine here through 10 weeks. But we also observed that a second shot of the vaccine given at 2 months substantially increased these titers by about 4-fold. So remember that number 4-fold, because I'm going to come back to it later. This vaccine also raised robust CD4 as well as CD8 T cell responses in the majority of individuals very quickly within 2 weeks. These data then led to the initiation of the pivotal phase 3 study to look at safety and efficacy of this vaccine. This was COV-3001, a phase 3 randomized double-blind placebo-controlled trial of the efficacy and safety of Ad26-CoV-2S. I'll say that the plan up front was to test both a single shot and a 2-shot version of this vaccine. The single shot efficacy study started in September. The 2-shot efficacy study could not start until November, which was 2 months later, because the FDA required data following the boost in the phase 1/2 program before they would allow the boosted phase 3 study to start. That's why there was the time difference. The single shot phase 3 study, COV-3001, was enrolled between September and December of 2020. 44,000 people were enrolled in many sites throughout the world with a 1-to-1 vaccine placebo randomization. This study tested the single shot vaccine at the dose of 5 × 10⁻10 viral particles. And this study was really led jointly by J&J together with the NIAID CoVPN network. The study was conducted simultaneously in 3 different regions of the world, in the United States, in South Africa, and in Latin America, including Brazil. So something extraordinary happened in the context of this phase 3 study, which was the emergence of variants of concern. Early on in 2020, there was a single point mutation that emerged from the original Wuhan virus. That was a D614G substitution. But we didn't really hear too much about variants until about this time last year, when we heard the emergence of multiple different variants, including the B117 variant, which has become known as Alpha, and the B1351 variant, which has become known as Beta. These variants have properties of either enhanced transmissibility or reduced sensitivity to antibodies, including vaccine-induced antibodies. In South Africa, we saw a very striking SEER epidemiology. The study was designed here and started in September 2020. During the enrollment phase of the study, there was essentially the near complete replacement of the virus population with the B1351 so-called Beta variant. And even to this day, of course, we don't know yet for Omicron, but up until now, the Beta variant is the variant that most effectively evades neutralizing antibodies to all the vaccines by essentially 4 to about 10-fold. So we designed a vaccine here, and it was tested against this Beta virus, essentially. So the efficacy collection period is shown in this rectangle, and essentially it was a test against a 95% population of the Beta B1351 virus. So this study was conducted in the setting of very challenging epidemiology. It was conducted in multiple geographic areas, including in the developing world, at the time of the peak global surge in December, January of 2020, 2021. including in South Africa, where over 95% of the virus was the B1351 variant. Nevertheless, we saw fairly good protection against symptomatic COVID infection, 72% in the US, 68% in Latin America, and 64% in South Africa, as well as a higher degree of 85% protection against severe disease. And this is the Kaplan-Meier curve of protection against severe disease. The placebo curve is shown in orange and the vaccine curve is shown in blue. And interestingly, you can see that the separation of these curves occur very early, even by day 7, which is interesting because this is really before there's any measurable neutralizing antibody response. So the immunologic mechanisms of this very early protection is really not clear. These efficacy data then led to FDA emergency use authorization on February 27th of the J&J vaccine, which is the third COVID vaccine. By that time, the Pfizer and the Moderna vaccines, both extraordinarily successful vaccines and extraordinarily effective, were already being rolled out to millions or tens of millions of people by then. But we felt that this vaccine had several unique aspects and unique properties, including the ability to reach hard-to-reach populations, and would complement the mRNA vaccines in the United States, and of course would have a very important role throughout the world. The rollout was not without hurdles, and in April there was a 10-day pause of distribution of the J&J vaccine because there was the identification of very rare but potentially severe episodes of thrombosis. The goal of the pause was to do a deep dive to ensure that this was a very rare effect, and this wasn't just the tip of the iceberg. We wanted to make sure there weren't hundreds or thousands of these cases that we weren't aware of. Thankfully, after a deep analysis, it was concluded this is an exceedingly rare effect. Approximately 2 in a million cases is the current numbers from the CDC. And the pause also allowed time to alert the healthcare community for the appropriate treatment of this thrombosis, which involves non-heparin anticoagulants because this thrombosis includes platelet factor 4 positivity. In addition, it allowed the CDC to do a very careful risk-benefit analysis, and the independent risk-benefit analysis demonstrated tremendous benefit, higher than risk for all subgroups, including in the subgroups most susceptible to thrombosis. And then after 10 days, then the role of this vaccine was resumed without any restrictions. So we were asking the question, as a single shot vaccine, what would be the durability? Would it be essentially insufficient to cause durable immune responses, or would the durability actually look quite good? So our first cohort of the phase 1 participants that we had at Beth Israel Deaconess Medical Center in Boston, we followed for a period of 8 months. We're actually literally now we're doing the 12-month immunogenicity data. But in July, we published the 8-month durability data for the Ad26-CoV2S vaccine. And here are binding antibody titers on the left top, neutralizing antibody titers on the right top, and CD8 and CD4 T-cell responses on the bottom. And we observed that this vaccine, even as a single shot vaccine, generated very durable responses. In fact, compared with peak immunity, 8 months later, there was only a 2-fold decline of antibody titers and essentially no detectable decline of T-cell responses. So this is actually quite remarkable durability for a vaccine. There were 3 individuals in this group of 20 that were outliers that had sharp increases of antibody responses. 1 was a COVID breakthrough case, and 2 received a boost with mRNA vaccines after the Pfizer and Moderna vaccines were approved and before the J&J vaccine was approved. So these data really show 2 things. First, that the vaccine is very durable, and second, that it also primes very well for any subsequent antigen exposure. We also looked at neutralizing antibody titers induced by the vaccine. And on day 29, which we previously thought was peak immunity, there were moderate titers against the vaccine strain, the ancestral Washington 1 2020 strain. But these titers dropped off quite significantly to various variants, including almost no detectable responses to the B1351 beta variant in South Africa, which was curious because we already had phase 3 efficacy data in humans showing that the vaccine provided fairly good clinical efficacy, and yet essentially no neutralizing antibodies. So it really suggests that immune responses other than neutralizing antibodies are key for protection. But there was also nearly a 10-fold decrease of neutralizing antibody titers to the gamma variant shown here and the delta variant shown here. But over time, something changed. Over time, even in the absence of boosting, then if we look 8 months later, then the responses to the vaccine strain, the Washington 1 2020, were only about 2-fold lower, but the responses to the variants actually appear to come up over time, suggesting that B-cell maturation process over time, even in the absence of boosting, that's not very well described. So we next looked at the kinetics of the immune responses of the 3 major vaccines in the United States: the Pfizer vaccine, the Moderna vaccine, and the J&J vaccine. And these data were published about a month ago. So here we compare the antibody responses induced by the Pfizer vaccine in blue, the Moderna vaccine in green, and the J&J vaccine in black, with 3 assays: a live virus neutralization assay that was performed blinded by Ralph Barouch's group at University of North Carolina, and that's complemented by a pseudovirus neutralization assay and an ELISA done here in our labs. But basically, all 3 assays gave very similar results. The 2 mRNA vaccines had astonishingly high antibody titers at peak, essentially 2 weeks after the second shot. The Moderna titers were nearly 6,000, according to the live virus neutralization assay, and the Pfizer titers were nearly 2,000. So incredibly high antibody titers several weeks after the second shot. However, at 6 months, these titers declined sharply by about a log, and by 8 months, by almost another log. So 8 months later, then the antibody titers had declined 34 to 44-fold compared with peak titers. And these titers declined to levels well below 100, which were identified in the Moderna phase 3 study as the titers likely needed for protection. So what we've seen with the mRNA vaccines is very high initial antibody titers that decline sharply and quickly over time. For the J&J vaccine, we see a very different kinetic picture. The antibody titers on day 29 following immunization, which we used to call peak, actually were much, much lower than for the mRNA vaccines, more than 10-fold lower, actually. But these titers were very stable over time, according to all 3 assays. So that at month 8, if we look at the live virus neutralization assay, the pseudovirus neutralization assay, or even the ELISA, then at month 8, then the antibody responses are more or less comparable across these 3 different vaccines. The Moderna titers are always a bit higher than the Pfizer titers, though. And if we look at different variants, we see the same pattern over and over again. Here we look at neutralizing antibody titers against the ancestral Washington strain, against the Delta variant, against the Alpha variant, and against the Beta variant. And we're really seeing the same pattern, which is that at peak, the 2 mRNA vaccines have very high titers, but these decline quickly and sharply. So the titers 6 and 8 months later are far reduced. With the J&J vaccine, after a single shot vaccine, the titers initially are far lower, but these appear to be stable over time or even go up over time. So if you look at the titers against Delta, which is the most common virus in the United States, at 8 months, then these titers are really quite equivalent. CD4 T-cell responses were seen with all the vaccines, and were generally quite stable over time. And CD8 T-cell responses, as you might expect, are substantially higher with the vector-based vaccine than the mRNA vaccines. So this study shows that, really consistent with much literature already, that mRNA vaccines induce very high initial antibody titers, but these responses decline sharply by 6 months and even further by 8 months. The Ad26-CoV2S vaccine induces lower initial antibody titers, but these responses show minimal decline for 8 months. And by 8 months, the antibody responses are relatively similar for the 3 different vaccines. Ad26-CoV2S also induces durable CD4 and CD8 T-cell responses. So how about real-world efficacy? The first real-world efficacy was seen in South Africa with the so-called Sisonke study. This was the first vaccine rolled out in South Africa in February. And so between February and May, investigators in South Africa vaccinated 500,000 healthcare workers and reported in August that this single shot vaccine provided about 90% protection against death and 70% protection against severe disease and hospitalization. So it really saved a lot of lives. And in the United States, there's many different real-world efficacy studies. I don't have time to discuss all of them. This is one of several. These numbers are sort of middle of the road compared with, there's some higher, some lower. This is based on insurance claims data in about 400,000 vaccinees and 1.5 million unvaccinated individuals from March to July. I believe now there's data through August. And this is corrected for under-recording of vaccinations. Without this statistical correction, the efficacy numbers would be about 10 points lower. But with this analysis, the efficacy was about 79 to 81%. And even if you look specifically in the later months, in states that had 95% or more Delta variant, the efficacy was very similar. So the efficacy was stable with really no evidence of decline, as shown here. So how about boosting? So these data are not from our group, but rather from J&J in the phase 1 and phase 2 programs. And they followed individuals for 6 months. And here, these data essentially have confirmed the data that we reported in just 20 people, showing very good durability over time for 6 months. And then they gave a boost at 6 months. And 1 week following the boost, then antibody titers were 9-fold higher than they were on day 29. And if you wait until 4 weeks, which is not shown here, it's 12-fold higher. So if you do a boost at 6 months, you get a 10-fold increase in antibody titers, which is much more than if you do the boost at 2 months, which I showed you earlier, and you get about a 3 to 4-fold increase in antibody titers. So you get more of a boost if you wait longer. We modeled this in monkeys, and we administered a single shot vaccine at the 2 different doses to monkeys. ELISA titers are shown on the top, neutralizing titers are shown on the bottom. There is some decline from peak, but generally stability over time. And we also looked at RBD-specific memory B cells in the monkeys, also showing durable memory B cells over time. If we look at the phenotypes of the B cells, then we notice that there's a shift from activated memory B cells in blue to resting memory B cells in orange over a period of about 6 months. And we speculate that the shift from activated to resting memory B cells underlies the improving boost that you get if you wait 6 months compared with 2 months. And so when we did boost these monkeys were at 8 to 10 months, we got an astonishing boost in binding antibodies shown on the left and neutralizing antibodies shown on the right. And when you boost, you actually boost very well to all the variants, which is similar to what's been reported for the mRNA vaccines. So this brings us to the boosted efficacy in humans. And the COVE 3009 phase 3 study essentially tested the efficacy of a 2-shot version of the vaccine. You can call it a single shot with a boost if you like. This enrolled 30,000 individuals, 1-to-1 vaccine placebo randomization that tested a 2-shot version of the vaccine with a 2-month interval. Now, I just showed you that a 6-month interval would likely be even better than the results here with 2 months. And this was an international study. The results of the 2-dose version of the vaccine showed 94% protection against any symptomatic disease in the United States. So essentially high efficacy that appears to match that achieved with the mRNA vaccines. It was lower at 75% globally, largely brought down by the Lambda and Mu variants in Latin America. And across the entire global study, 100% protection against severe disease, although numbers were low. So people have asked, should this vaccine be a 1-shot or a 2-shot vaccine? And I think the answer is both. If you're in a state such as in South Africa in February of 2020, and there's no other vaccines available, you can really calculate that a single shot vaccine with 70% efficacy saves a lot more lives than a 2-shot vaccine with 94% efficacy. The single shot vaccine leads to durable antibody and T-cell responses for over 8 months with real-world efficacy for over 6 months. The 2-shot version of the vaccine, however, provides high efficacy, 94% in the US, that matches peak efficacy reported with the mRNA vaccines. Mix and match regimens combining mRNA and Ad26 in either direction are also being explored and might have very interesting properties. Boosting at 6 months is more effective than 2 months, likely because of this phenotypic shift from activated to resting memory B cells. So I just provided a summary slide really showing the wonderful progress of vaccine development overall. These are just the results from the phase 3 randomized control trials from the major publications showing the 2-shot Pfizer vaccine with an efficacy of 95%, the 2-shot Moderna vaccine with an efficacy of 94%, and the J&J vaccine single-shot efficacy 72%, 2-shot efficacy 94%. And these have had tremendous impact both in the US and throughout the world. In the US, the Pfizer vaccine is used by far the most widely. Over 120 million people received it. 77 million people received the Moderna vaccine. A lower number, but still a large number of 15 million received the J&J vaccine. But in low-income countries is really reversed. As of last month, the mRNA vaccines have had relatively little penetrance to low-income countries. The J&J vaccine has distributed 25 million doses to low-income countries, and this should increase to several hundred million by next month. So I'll close with a few perspectives. Vaccine development for COVID-19 has proceeded faster than for any pathogen in history. Safety and public trust are critical. Currently, boosters are the current focus of much discussion. While they are important, I believe they are also somewhat distracting to the top priority, which in my opinion remains providing initial vaccines to the unvaccinated individuals in the US and all throughout the world. And the reason it's important to have a global perspective is because, and I actually wrote this statement long before this past weekend, new variants will likely continue to emerge in areas of the world with surging infections and low vaccine coverage. This happened with Delta, this happened with Omicron. And as long as we have large regions of the world that are poorly vaccinated, Africa currently has less than 7% vaccine coverage, then we should expect new variants to emerge that will come back and threaten the rich countries of the world as well. So I do feel that multiple vaccines need to be deployed as quickly as possible to accelerate the vaccine rollout in all regions of the world. I'm often asked, when will the pandemic end? I can't answer that. I've been wrong many times. But I do think it's really a race between how fast and how deep we can vaccinate the entire world and the emergence of variants that will come back and threaten us. So I'll close there. There's too many people to thank. The non-human primate studies were conducted in my group with collaboration from J&J, Biochemistry, and many other academic groups, including the Reagan Institute. And our clinical studies, our own clinical unit at BIDMC. This is, of course, only one of many, many sites of the overall clinical program. And this is a picture of our group from about 2 years ago, so horribly outdated. We were trying to get a new picture this summer, and during the few-week period where we didn't have indoor masking, but unfortunately, we were too late, and our picture was scheduled after indoor masking rules went back into effect, so we have to wait a little bit longer. But hopefully, with high vaccine coverage, we'll be able to take another picture again. Thank you very much.
Thank you, Dan, for a wonderful talk and a titanic scientific effort over these couple of years. I mean, it is an impressive amount of work and beautiful science with tremendous impact. I mean, congratulations for that. And
thank you.
And we have several questions, Dan. I mean, probably we will not have time for all of them, but one that really you mentioned in your perspectives and is something that people has in mind is, will these vaccines protect to this new variant Omicron? I mean, yesterday the CEO of Moderna expressed some concerns regarding their vaccines, and probably it's too early to say, but what will be your views there?
Well, we know very little about Omicron. We knew almost nothing about it last week, a week ago at this time. So it's very, very early days. And while I don't think we can answer your question today, what I can say is that there are many, many committed groups of scientists around the world that are going to intensively study Omicron. I think in the next 2 to 4 weeks, there's just going to be a deluge of information. How it's going to turn out is an empiric question that really needs experiments. So it really shows the importance of experimental science to answer questions in real time about what's happening in the pandemic. So I guess my hat's off to all the scientists throughout the world who are willing to give up time during their Thanksgiving vacation and temporarily put aside all the wonderful work they're doing to address this important problem. We are also part of that effort, and so many groups, including our own, over the next several weeks will test vaccine sera for binding and neutralizing antibodies, as well as T-cell responses against the Omicron variant. What I can say is worrisome is that it has over 50 mutations, including over 30 mutations in spike, and they're concentrated. There's over 12 mutations right in the receptor binding domain. And that's why people have become very worried, because there's a concentration of mutations, some of which are known to give partial resistance, some of which are unknown in terms of their effect, right in the target region of neutralizing antibodies. And so there is a widespread expectation there will be some hit to there'll be some level of escape, whether it's a manageable, relatively low level, or whether it's a very high level escape is not yet known. And of course, that empiric question is critical as to whether there will need to be a redesign of vaccines or if the current ones will work out just fine.
And Edward is asking a question that is relevant probably to the redesign or to an eventual future pandemic that may be not far away. And the question is, you mentioned you have gone and the community has gone very far to generate a vaccine in a year, but all this that we have learned could deliver in the future a faster vaccine for a new pandemia or a new variant?
I do think so. I think Pfizer has announced that if needed, they could have a new vaccine ready in 100 days, so in about 3 months. Now, of course, that assumes that you can just do a short-term immunogenicity study. You don't have to do a long-term efficacy study. J&J for the ad vector platform can also do it quite quickly, probably 1 or 2 months later than that. So I do think that there is the technology and the expertise and the know-how available to make vaccines even faster than before, particularly if it's a variant of a SARS-CoV-2 vaccine. If it's a new pathogen altogether, then I think all bets are up, and it kind of depends on the degree of confidence you have in immune correlates of protection. But ultimately, it's going to be much harder to distribute a new vaccine than it will be to make a new vaccine, because the logistics of distributing a vaccine, particularly to the developing world, is probably going to take a lot more time than it will be to make the vaccine itself. We haven't even been able to distribute the current vaccines to much of the developing world, and it's been over a year since they've been available.
A couple of questions of the audience that are very probably general but relevant is, what is special about Adeno26 and what made your choice at those early times to choose this as a vector for vaccines?
So we looked at, so the adenovirus, so first the adenovirus class was more manageable and more potent and more stable than many other classes of viruses, such as pox viruses and other classes of viruses. Within adenoviruses, at that time in 2002, when I started my lab, there were 2 large programs in the HIV field that had really developed adenovirus serotype 5 vectors. One was the program from Merck, and one was the program from the NIH Vaccine Research Center. So there was a lot that was known about adenovirus serotype 5 vectors, but we already knew that Ad5 vectors would be very limited, both in terms of immune phenotypes, in terms of exhausted T cells, as well as in terms of the fact that in the developing world, 50% of people have high neutralizing antibodies against the vector of over 1,000 and likely would be suppressive. So that's why we had a strategy to develop serotypes other than Ad5 for an adenovirus vector platform. And we looked initially at about a half dozen, and now we've looked at a total of more than 24 serotypes of adenoviruses from humans as well as non-human primates. And Ad26 is a very good balance of various technical parameters, such as relatively low seroprevalence, although it's not 0, but relatively low, so it's not a problem, together with high immunogenicity, and also genetic stability and manufacturing capacity and titers. So it has sort of a good balance of the immunologic as well as the practical properties for vaccines.
Probably last question then, because we are running out of time. I mean, all vaccines are administered or compared on single. Now people is given the option to get this mixed cocktail of vaccines. I mean, are there studies in analyzing this, and what would be your views on that?
So the NIH has a study looking at the different the so-called mix and match combination. I mean, we meaning our group and the field has been studying what we used to call heterologous prime boost regimens for over 20 years. Now it's being sort of reinvented and called mix and match, which is actually probably easier to say. But there's a lot of knowledge in animals and in humans about combining different vaccine modalities. And the concept is that different vaccine modalities might bring different immune spaces. And you might be able to sample different immune spaces, and you might be able to get the best of both worlds if you combine different vaccine modalities. I personally think it's quite an interesting idea. And so what's being studied is Ad26 followed by mRNA, as well as mRNA followed by Ad26. At BIDMC, we're doing a study now. We actually have an initial data set that will be ready to publicize very, very soon, in which we have enrolled individuals that originally received the Pfizer vaccine, and they either were boosted with the J&J vaccine or with a third shot of Pfizer. And it's showing some very, very interesting immunologic differences depending on whether you get a homologous versus a heterologous boost. So that's to come.
And again, thank you so much for your time in such a busy time for you and your lab and society. So thank you so much for a beautiful talk. And for
thank you very much, Facundo.